Now I would like to request the Ms. Pooja to introduce the next speaker.
Dr. Sugunakar is currently working as assistant professor at Lovely Professional University, Punjab. Dr. Sugunakar works on the clinical cancer genetics, cancer epidemiology, and various cancer types. As part of his PhD, he worked on a topic entitled FLT3 mutation screening and SNP analysis of NHEG repair and extrinsic apoptosis pathway in acute leukemia, which involves the screening of low penetrating and oncogenic mutations in the development and progression of disease by studying the genotype associations based on epidemiological and clinical parameters in the origin of acute leukemia. He also has 7 years of teaching experience for postgraduate and graduate students at Department of Genetics and Biotechnology, Osmania University. He has published 22 papers and 3 book chapters with H-Index 10, trained 50 plus master students. He also is also awarded the Best Poster Presentation Indian Microbiology Association, Best Poster Presentation from INBIX 2017 Jaipur, Best Teacher from Joginipalli BR Engineering College, RR District. I request Dr. Sugunakar to take upon the session. Sir, please.
Is I'm audible?
Yeah, yes, sir, you are
audible, and it's a gentle request from team InSyB. Please try to finish in between 15 to 20 minutes. Please try to understand. Yeah, thank you. Thanks for understanding.
Yeah, even I have a meeting at 12:40 at the university, so I'll try to see how can I justify that. Yeah, thank you very much. First, I would like to thank the organizers, especially my guru. My professor, my big mentor, whose support was immense, that is Professor P. Kaviteshwar. Way back to 2001, 2003, he is my first coordinator for biotechnology when I was doing my MSc. So a big thanks to you, sir, for allowing me to speak something on what work I have done. Thank you so much, sir. And I would like to thank Dr. Jalaja, Dr. Anil, and Rangarao Ambati for giving the opportunity, and also the Vignan institutions. Thank you. Yeah, my topic would today I'll be speaking mostly on Envision, the role of cancer prognostic biomarkers to intervention of bioinformatics. Okay. Yeah, the presentation would discuss under these segments. One is on clinical front, prognostic biomarkers, ongoing research, which I'm doing. Yeah, I'd like to thank Kenta Nakai yesterday for a briefing about the disease-associated variants in different categories, which was highlighted in BMC genomics and also in cancer translational medicine, where we are trying to look at, as a part of my discussion, I'll be mostly talking on cancer somatic mutations and with regarding to different diseases, especially with regard to acute leukemia. Yeah, so when of disease-associated variants that we come across is Mendelian disease variant, cancer predisposing germline variants, and recurrent cancer somatic mutations are more serious than that of the complex disease variants. So the background of the study that I've taken is the leukemia. Leukemia is a group of heterological hematological malignancies, which is caused because of the chromosome abnormalities. Presence of immature white blood cells, they're nothing but the blast cells. This is one of the important prognostic marker when you talk about the poor response. And the progression of the disease of acute leukemia is basically depends upon the percentage blast. Absence of mature white blood cells leads to anemia, thrombocytopenia, and leukocytosis. If we can able to see this particular schematic view, we can able to understand that there is a transformation of a normal hematopoietic lineage to an abnormal development that leads to acute leukemia. And the transformation may be because of the dysregulation of cell surveillance mechanism, that is with regard to the genomic instability and also the apoptosis. Okay, the Abnormalities include dysregulated DNA repair, failure of cell cycle checkpoints, failure of apoptosis, activation of oncogenes, and failure of tumor suppressant genes. The clinical symptoms like fatigue, shortness of breath, on exertion, easy breathing, pitachi, and other commodities. When we talk about the classification, it all depends upon the rate at which it is progressing, and it is further classified into, the leukemia is further classified into acute and chronic leukemia. I'll be mostly talking about our acute leukemia. It is an abrupt onset of severe disease with immature cell forms of both ALL and AML. When you talk about the hypothesis of the study, when you talk about the hematopoietic stem cells taken into consideration, they are something called as they are lifelong survivors which are prone to damage by various exposures of exogenous as well as endogenous factors. ATM is a sensing gene. They're called as DNA sensors. Okay, when some damage is happening, the first damage is responded by a group of genes, that is ATM and ATR complexes, and this particular ATM will sense the DNA damage and activates the downstream repair pathways. And to the surprise, NHEJ pathway is the first pathway to get activated in this hematopoietic lineage. Since it is an error-prone pathway, dysregulated NHEJ mechanism might lead to genomic instability and leukemogenesis. The imbalance between the proliferation and the apoptosis associated with the differentiation arrest, it results in survival and clonal selection of the leukemic stem cells, which might contribute to chemoresistance among the patients. The failure of apoptosis and dysregulated error-prone NHEJ pathway exert influence in, it can be individually or it can be cumulative impact. Now, as a part of the study, we have collected samples from MM-GBSA Cancer Hospital and Institute of Medical Sciences. The total sample size is 772 sample size, and out of which the acute leukemia are 446, which comprises of 225 AML and 221 ALL cases, with a control population of 326 samples. Informed consent was taken from all the subjects, and the inclusion criteria, the primary de novo samples have been taken in the study with a confirmed diagnosis based upon the CBP, bone marrow aspiration, and cytogenetic analysis. Exclusion secondary leukemia and therapy was excluded. Ethics committee approval was being taken by both the hospitals, and the epidemiological and the clinical information was obtained from the tumor registry and also with the help of the medical oncologist. Now, the methodology broadly divided into experimental analysis, then we have statistical analysis, then followed by in silico based approaches. I'm skipping this because I don't want to get into in those things. Now, when we talk about the epidemiological and clinical variables, we try and see that when we talk about the age at onset with regarding to the AML and ALL, there is a slight difference between both of the both these disease types. AML basically happens in the elder age group, and ALL happens in the younger age group. You can able to see here clearly that in the group of 20 to 30 years and more than 30 years, the percentage was 34.2 and 44.9 when compared to the ALL patients less than 10 years, 30.8, and in the age group of 10 to 20 years, 42.1%. And there is an increased elevated risk of male preponderance in developing acute lymphoblastic leukemia in males when compared to the females. It may be because of the exposure. And when we talk about the area of living, the rural population are more prone to the diseases in both AML and ALL, 65.3 and 57.5. It is because of the exposure to the pesticides in the agricultural fields. And with regard to the habitat, we can able to see a double risk of AML patients when compared to the ALL patients, and benzene is one compound which is an important compound in the development of the acute leukemia. And further, we try and talk about the mean SD levels and baseline clinical characteristics at diagnosis. We try to see here that already I've discussed that age at onset, the mean SD at age at onset is 30.88, okay, when compared to the 16.81, and these are the characteristic features that AML is an elder age group when compared to the ALL. And further, the peripheral blast, which I've talked about, the immature blast cells, which are the prerequisite for development of acute leukemia, and the presence of blast cells, which indicate it's a poor prognostic marker, and this shows the poor progression with regard to the disease. And the response rate will be very less when you see high blast percentage. Furthermore, the WBC count, the platelet count, LDH, and disease-free survival have seen a significant increase when compared to the control patients. And now further, how we have the study, basically we do it with epidemiological, clinical, and molecular parameters in the development of disease. As a part of my study, I've taken NHG repair pathway, including the ATM and also NHG repair pathways, along with extensive apoptotic pathway. We have completely studied the complete set of SNPs which are present in both the pathways, and that's where the imbalance has been seen with regard to these two. So we have selected a set of SNPs that are present in the Fas receptor, Fas ligand, caspase 8, caspase 3, and caspase 7. And further, to understand its statistical significance, we further underwent to do the interaction studies. A lot of slides have been skipped in the brevity of time. Then we talk about gene-gene interaction studies. There's something called as linkage disequilibrium, multidimensional reduction, combined genotype analysis was being performed. When you have done that linkage disequilibrium analysis, the LD results, that what we got from our results indicated that the specific genotypes of NHG, ATM, and extensive apoptotic pathways may confer risk independently. Because they're present in the different chromosomes, as the alleles are present on different chromosomes, they segregate individually. So that's where we concluded our result that it is both implies to both AML and also ALL. Further, the multi-dimensional reduction analysis has been done, where we have seen the results saying that they always act independently, and it also shown that it has gotten a synergistic interactions between these SNPs of XRCC6, okay, minus 61 C2G and minus 1310 C2G polymorphism may be possibly having an epistatic nature and observed in haplotype analysis. I've skipped that particular haplotype analysis part because of time, where in the presence of two variant alleles conferred protection. When the two variant alleles are present, it is conferred protection, but when each variant allele is present, it is conferring risk. It is conferring significant risk of development. This may be because of the variant allele at one locus may be suppressing the other allele. That's where the concept of epistatic has come into the picture. And the variant allele leading to a normal expression of XRCC6 gene. While the variant SNPs individually might be associated with the NHEJ activity. Individually, they're having a risk, but when they're combined, they're showing a protective nature in the normal XRCC6. However, the conclusions have to be confirmed by in silico, in vitro studies using cell lines and other studies. Now, this is regarding to the ALL cases. The MRD analysis have revealed the influence of NHEJ and apoptotic pathway genes in the origin of ALL. So the synergistic interactions between XRC6 SNPs was evident in both AML and ALL, but the effect of apoptotic genes of ALL was found to be high. The results reveals the lineage specific variations between AML and ALL, AML susceptibility may be one of the factors. Further, we done combined genotype analysis where we have taken the cumulative adverse genotypes and we have done the analysis and try to see, we've done the odds ratios and try to see how they are reacting. So we have performed an independent analysis of SNPs in both NHCJ, ATM, and also the extensive apoptotic pathway. It has shown a significant association of XRCC5 with regard to the VNTR polymorphism, with regard to the XRCC5 6 SNPs, with regard to the FAS genes, and also caspases. Okay, with regard to the, one more caspase, it's not only insertion and deletion was taken with regard to the AML. We have seen a significant association between the risk genotypes among the different genotypes in both AML and ALL cases. The same thing was being observed with regarding to the acute lymphoblastic leukemia also. There was a linear increase in odds ratio with respect to the number of risk genotypes in both AML and as well as ALL. There is 5 risk genotypes were taken to taken to the study, which results that a cumulative effect of risk genotypes of NHCG and ATM and extensive apoptotic pathway on acute leukemia development. So there is an association of risk genotypes. When they're combined together, the adverse genotypes will facilitate in the development of acute leukemia. It may be possible that dysregulated NHCG repair in combination with the low apoptotic ability may predispose to acute leukemia. The genes involved in these pathways are low penetrating genes and may contribute small but cumulative effect if taken together. Now, these are the genotype and phenotype correlation studies that we have performed with regard to the SNP, their location, and the risk allele that is being developed, the genotype, and what is the functional significance. If you can able to see, ATM has got reduced DNA repair capacity, and XRCC has got enhanced DNA repair capacity. These are part of, when you have done our studies, we come to conclusions that there are some significant elevations with regard to each and every thing that is taken into consideration. And when we see the association with regard to the AML and ALL, with regard to the complete remission and the DFS, disease-free survival, there was a reduced expression of this particular genes has been taking place. And before we started doing this analysis, we have done our in silico analysis with regard to the protein-protein interaction studies. It has revealed an interaction between the genes what we have taken. This work was done along with Dr. Prashant at BSIR and Mohan Lata, who are gracious enough to help me in the initial time when we were trying to understand that, when we were trying to develop this hypothesis. This particular image has really helped us to move forward with a very positive note. And further, we have validated the study with regard to the ASPE analysis, with regard to the SNPs that were taken. And further, based upon the classification of the SNPs, based upon their location, we have done different in silico analysis with regard to the SNP location, SMP location in the promoter region, intronic region, and the exonic region. So we have used different softwares. So, for example, as a part of that, we have used a transfer data database from the bio-based biological database called as Alibaba 2.1 version. By using a transfec, the presence of a variant allele, you can able to clearly see that. And ATM minus 544, A to T polymorphism. A is converted into T. So there is a variant allele that is coming to picture. The presence of an A allele is giving rise to an SP1, which is giving rise to a series of transcriptional factors binding. But the presence of T allele, you can able to see that there is a new transcriptional factor which has come. Means the variation of a single nucleotide also has an impact on the transcriptional factor binding efficiency. In the inference, says like, it revealed the presence of T allele resulted in gain of site of transcriptional factor, which may act as a negative regulator of gene expression, which may act as a negative regulator. Okay. Further, we have studied with regard to the XRCC201 VNTR, then XR6-61C2G, then 1310 C2G polymorphisms. We have been able to see that the presence of this particular, when the variant allele is changing, there's a change in the transcriptional factor binding efficiencies, or there's a loss or the gain of transcriptional factors which are highly regulated, and by which these are leading to the disease progression. And further, we combined with regarding to the results by using thermodynamical ensemble predictions. We have done analysis with regard to the XR6 C2G and 1310 C2G polymorphism. We have further done the ssDNA sequence of these two SNPs and the impact of the rate of transcription with regard to the GGG insertion in the promoter region associated with more negative energy may confer stability leading to the optimum expression. This explains the result of haplotype analysis, which revealed that reduced risk of GGG genotype to develop acute leukemia through individually. That's what we try to show in the linkage disequilibrium, in the statistical analysis, and as well as in the haplotype analysis, and also with regard to diplotype analysis. They are conferring risk independently, and it has got an impact on the epistatic impact is also been present. In the list of SNPs that are taken and the factor binding site, we can able to see that these are the different SNPs that are taken into consideration, and the presence of this particular associated allele, it is showing different kind of transcriptional factors. So there is always, if you have a for example, you have Fas receptor, if this particular SNP is present, there is a loss of HSC binding site. Okay, there is a gain of site, there is a loss of site, because of which there is an altered expression of gene expression has been noticed. Now further, we have done the intronic analysis, XRCC5 240 G2A polymorphism. We also seen that there is an aberration of the intronic ESS site, which has gotten a potential altering ability. So these are the list of different intronic regions that has been taken as a part of the study, and it has shown that there are some potential splice site has also been created, and it also impact on the sequence. Then we further done EXONIC analysis, and we have seen the energy levels between a normal SNP and also an abnormal normal allele, and also an abnormal the variant allele. We could able to see a significant energy levels which has been seen in both the C allele and as well as in the G allele and also the C allele. Exhibited around the free energies were calculated. Okay, and we see that hence the stable mutant RNA may interfere with the wild type during the protein synthesis, thereby resulting in the altered protein levels. So that is what been observed. Now, as a part of the study, we have contributed, we significantly can contribute, and we have recommended that a mandatory screening of FLT3-ITD, which is one of the oncogenic mutation, which I have not discussed in the brevity of time. It's a mandatory screening of FLT3-ITD. The presence of this particular FLT3-ITD mutation, the patients will not respond to chemotherapy. So it is when we see in Indian population, in the South Indian population especially, we have seen 18.7% of this particular patients what we have collected in the hospitals, out of which 18% out of for example 226 samples what we have taken, out of which 18.74% have shown FLT3-ITD mutation, and they have to go for different kind of personalized chemotherapy is being given to those particular patients. So presence of FLT3-ITD is very crucial in responding to the chemotherapy. Further, Fas ligand can be an alternative therapeutic target, and ATM XRCC5-6 can be used as biomarkers in the management of acute leukemia. This is first part of study. Then we talked about something called as a prognostic biomarkers. We have different cancer types where we have taken 2 examples. I have a lot of other stuff. Some of the papers have been highlighted what we have worked on. We have seen in the breast cancer and also in acute leukemia. In breast cancer, we have tried to target this particular mTOR. The PDB ID is 5H64, which is a mammalian target of rapamycin, mTOR kinase, which is an intracellular signaling pathway, which basically helps the cell survival mechanism, the cellular survival, in the cellular morphology, and as well as in the protein synthesis and integration of metabolism. There are many factors that this particular, many downstream pathway, this particular mTOR plays an important role. The dysregulation of this mTOR pathway, including breast cancer and its hyperactivation, commonly associated with cell growth and rapid proliferation and neogenesis. So the dysregulation has been caused to that. When you try to see with regard to acute leukemia, as I already talked about the somatic nature of this particular cancer, we try to see isocitrate dehydrogenase, which is one of the important catalyze enzyme, which helps in the oxidative decarboxylation. It mutated version, which will lead to oncometabolite production. That several it disrupts several downstream molecules, which basically undergoes the blockage of the pathway that's taken into consideration. So inhibition of this mTOR and as well as the IDH, mTOR with regard to the breast cancer and also the acute myeloid leukemia. We have taken two different, there are important prognostic markers which have been present as a part of the cancer therapy. These are very important. So as a part of this particular study, we have done some in silico based analysis by using the molecular docking and molecular dynamics analysis with regarding to both breast cancer and acute leukemia. We have used different kind of inhibitor molecules, which has been taken from the PubChem database. We have used almost a similar kind of protocols for both of the things. And we used a different kind of drug molecules. And this is how we have selected the molecules. The system starting from the suite what have taken by Schrodinger drug discovery suite has been taken, then selection of the protein targets and the known inhibitors were taken into consideration. And we have taken around 40 compounds which selectively consider the best anchored compound, then preparation of the protein, lead compounds and the database preparations, and active site predictions and receptor grid generations have been done. Furthermore, by molecular docking, high throughput virtual screening is being done, and further done with your boiled egg plot analysis. Now, these are the results that we have seen that the compound Genotype 855694445 showed it has a high affinity with the mTOR protein. And further, we have studied the upstream and the downstream analysis with regard to the hydrogen bonding interactions, the electrostatic interactions, then aromatic interactions with regard to which particular amino acids have been targeted and how it is binding. Then we have studied the with regard to the active site with regard to the mTOR protein. And then we have done the boiled egg plot predicting the best 3 compounds to establish drugs by using virtual screening drugs that we have taken into consideration. The lead compound, Genotype 855694445, is an ideal compound for pharmacophore profiling. The compound shows the best affinity among the pre-exist drugs and inhibited drug against the target mTOR, as well as the study of pharmacophore profiling is also an important criteria. We will encourage, like we encourage to do further analysis with regard to this particular mTOR to prevent the breast cancer, and trying to see how it is going to subside the other pathway analysis. And this is with regard to the IDH of acute myeloid leukemia. The same analysis has been done. We have seen there is one compound called as SHEM-BL1639 1748 in the PubChem database. We have seen that effective drugs which are basically combining and trying to inhibit the activity of the IDH dysregulated protein. We come across the different binding efficiency studies, and further we got the results. We got in a very promising results, and we are planning to consider for our downstream work with regard to the treatment in AML in the future. And presently, we are undergoing ongoing studies with regard to the erythrosis and the prostate cancer, and also the phylogenetic insights into the diversity of human papillomavirus in cervical cancer. This is in collaboration with Dr. Prashant Suravarjula. My PhD student, Dr. Ms. Bhumandeep, is working with Prashant and myself. And we're basically looking at the erythrosis and how it is related with prostate cancer development. We have tried to do some correlation studies with the bioinformatics approach. We have enlisted around published, we have taken enlisted the published papers of erythrotic genes through PubMed gene database, and further analysis is being done. We are trying to see what are the further analysis that we can plan for validation. And as a part of that, we've done some PPA interaction studies where we have tried to see that 22 erythrosis and other 20 associated genes of Homo sapiens using GeneMania and string database was done. It has shown some co-expression of about 51.29 and collocations of 30.59, and then predicted as 8.4 and shared protein domains of 4.79. So the work is still has to undergo, and this is one more phylogenetic insights that we have tried to see with regarding to the HPV cancer with regard to the cervical patients, where we have collected some data from the NCBI. We have retrieved CDS sequences from NCBI database, and then we have performed some phylogenetic analysis. Still the work is ongoing, and we have done we try to see what are the high risk and low risk genotypes which have been present as a part of the study. We have done some just a small initial work, and further analysis has to be done. Yeah, I always love this particular quote. The more I study, the more I learn and absorb, the more I realize how truly little I know. But Steve Marabola, thank you. This is from LPU, and I thank the LPU for giving this opportunity and to allow me to do my work. And I would like to thank the big list of people present. My PhD mentor, like Professor S. Vishnupriya, then my medical oncologist, my co-guide, Dr. Raghunath Rao, and the gynecologist. Right now, my gynecology, cervical cancer, I'm working with Dr. Leela Digumurthy and Sudha Sinha at M&J Cancer Hospital. Dr. M. Raghunath, where I did my ICMR postdoc at NIN. He was very kind enough to help me in my analysis. Dr. K. Rajendra Rao from NIN. My collaborators, Prashant, Prash, who is always instrumental enough in helping me in most of the analysis what I'm trying to do. Basically, I'm a wet lab scientist, but when it comes to bioinformatics, I only always look up to Prashant and trying to learn a lot of things from him. And my senior, Dr. Naishara Abdullah, who is at Shastra University, Dr. Shailaja, she's scientist at KIMS. Sandhya Amanani, my classmate during my MSc, and Smita Sipawar, the head associate professor at Department of Genetics. My lab mates, Dr. Anuradha, Manjula, Santoshi Rani, Prajita Fani, and Dr. Padala. And above all, what are the work that I have done till today with regard to the clinical cancer genetics? It's all because of the patients who has contributed to do some kind of analysis and trying to help the future prospects where we can really help someone who's really in need. And my PhD scholars Santoshi Kumar Dupala, Bhumandeep Kaur, Sadbir, and Pravalika Ala, they're currently working under my guidance. Yeah, thank you.
Thank you, sir. Thank you so much for the insightful talk. And I would like to thank all the speakers who have carried out and delivered their talks in the technical session 3. With this, I conclude this technical session 3 and commence technical session 4.